Hello and welcome back to Exercise Academy. So in this topic, we have talked about the stem cells. Okay, like what are stem cells and how stem cells are helping in the cell regeneration. Okay, so till now we discussed about the different types of epithelial skin, epithelial stem cell, epidermal, hair follicle stem cells. Then last time we also discussed about something called bulge, in which hair follicle is present, and we have got these sebaceous gland stem cells, epidermal stem cells, and the hair follicle stem cells. Along with that, we also discussed about the intestinal epithelium stem cells as well as the skeletal muscle stem cells. Okay, now today just let's talk about the medical applications of the adult stem cells. So we know that the stem cells are there which has got the ability to repair the damaged tissue, right? And if these stem cells could be isolated and propagated in culture, so actually this is possible. It is possible. The only thing is the maintenance is very high demanding. They could be in principle used to replace damaged tissue and treat a variety of disorders like diabetes or Alzheimer or Parkinson's. The use of embryonic or induced pluripotent stem cells is likely to provide more versatile approach to treatment of a wider variety of disorders. So in this lecture, we'll discuss like how these stem cells, okay, these stem cells will be used for the treatment of these particular diseases. So even though, see, we know that so much of science and technology is there, so much of knowledge is there, research has been done, but yet we are not able to cure this Parkinson and Alzheimer. So there is no perfect cure till now. Now, why this is not possible, we'll also see when we'll discuss about the therapy. Okay, so a well-established clinical application of adult stem cell is hematopoietic stem cell transplantation, that is bone marrow transplantation. So this is actually possible and it has been successful also. So for example, but this is only possible in the young patients, like where they are in their development phase. If you cross 40 and 50 and then you go for bone marrow transplantation, then it is not going to work properly. So it's all because of the aging, right? Now, if this factor is not there and you are in a young healthy organism, okay, so from a healthy donor to a healthy recipient, if you give, then it is possible. And the treatment of a variety of cancer is only possible, like you can say that that person is cured from cancer only when if it is in the first and second stage. If it crosses to the third and fourth stage, then it becomes difficult for any therapy to treat for the cancer. Okay, Chinese stop me in between if you don't able to follow. Okay, now most cancers, they are treated by chemotherapy with drugs which will kill rapidly the dividing cells. So dividing cells, so we know that in the case of the cancer, they are rapidly proliferating, right? So what they're going to do, this chemotherapy drugs, they actually damage the DNA or they inhibit the DNA replication. Okay, so the only problem with this is that it does not, okay, chemotherapy does not specifically kill only the cancer cells. It is killing all the dividing cells. So in normal case also, the normal tissues, the normal cell which is there, right, even they are getting damage, even they are getting killed. Okay, that's why you see that in the case of the cancer patients, in the case of the cancer patient, there is hair loss is there, right? Because this chemotherapy not only targeting the cancer cells, but it is also targeting your normal cells as well. The hematopoietic stem cells are among the most rapidly dividing cells of the body. So, the toxic effects of the anti-cancer drugs on these cells frequently limit the effectiveness of the chemotherapy in the cancer treatment. And of course, the new RBC stops forming. So, because of that, the health of the patient is deteriorating. Now, hematopoietic stem cell transplantation, it provides an approach to bypass this toxicity, thereby allowing the use of higher drug doses to treat the patient's cancer more effectively. So, in this procedure, the patient is treated with high dose of chemotherapy that would normally not be able to tolerate because of the toxic effects of the effects on the hematopoietic system. So, what we're saying here is that we are giving them the toxic doses of the chemotherapy. Okay, So what will happen? The potentially lethal damage is repaired. However, by transferring the new hematopoietic stem cells, which is taken from the bone marrow peripheral blood to the patient following the completion of chemotherapy, so that the normal hematopoietic system is restored. Did you get it? So what we are doing over here is that in this hematopoietic stem cell therapy, we are giving high doses of the chemotherapy. Okay, so this drug high dose is going to kill the entire hematopoietic system of the patient. Now what we are going to do is we are going to provide them the new set of bone marrow or these cells from that, so that the new hematopoietic system is restored. Did you get it, Chandni, over here? See, what we are saying here is in these hematopoietic stem cells transplantation, so here is your cancer patient, okay, the tumor or any kind of cancer is there, so you're giving high dose of chemotherapy. So what will happen in this case? So this high dose of chemotherapy, not only the cancer cell, but also the normal system it is going to kill, right? So the drugs are killing the tumor cells as well as it is toxic to the hematopoietic system. Now, once the existing hematopoietic system of the patient is gone, okay, it is toxic, it means even this is gone. What you're going to do is you're going to introduce new from the donor, okay, to the patient. So that is your hematopoietic stem cell transplant. So either the bone marrow you're going to transfer. Now, this new hematopoietic system, which is being taken from the donor, it is going to get restored. And that's how you actually kill the tumor cells. Okay? Krishita, clear?
Yes, ma'am.
Yeah. So, in some cases, the stem cells are obtained from the patient prior to chemotherapy stored and then returned to the patient once chemotherapy is completed. Now, however, we have to ensure that these cells are not contaminated with the other cancer cells. Otherwise, कोई मतलब नहीं हुआ, ठीक है? By taking from the donor and giving to the recipient. So in addition to their use in cancer treatment, these stem cells, they are transferred to the patients to treat other hematopoietic disorders like, immunodeficiency or the hemoglobin disorders. Now let's talk about the pluripotent stem cells, cellular reprogramming and regenerative medicines. Okay, before we proceed, any idea why most of the stem cell therapy is failing? It's not that effective, like out of 100 it is only possible for 30 to 40 cases. Any idea why most of these stem cell therapies are not able to, they are not successful? Any idea why?
Ma'am, is it because of the specificity, body not able to accept?
Yes, that is also one of the reason. That's true. And
No idea, ma'am.
okay, see, it's not that we don't have, right, in India or any other places we do have stem cell therapy going on. It's just that how easily we say, right, taking extracting the bone marrow from one person and giving to the other person, it is not that easy procedure. It is like, it's very painful procedure. So that is like one, as you said, is all because of the specificity. Correct, it is there. And one more thing is that every time when these procedures are being taken, there is no 100% guarantee that this is going to be a successful, because these are generalized. It's not like, how should I say, every patient is going to respond differently, है ना? So sometimes even there is a rejection. Sometimes the chemotherapy dose, like as we just discussed, that the chemotherapy dose first we should give high toxic dose, right? If the patient is weak, at first stage itself the patient might die. right? Even that is possible. The second, they have shown like this, like this, giving the stem cell therapy and all. It is not like this actually. So you actually make a hole in your bone, femur bone, you make it and you extract the bone marrow from there, and then you transplant to the patient. And this is all done when the patient is not it's not given the anesthesia. So just imagine how much painful procedure it is for both the recipient as well as for the donor, right? So all these cases are there where if you read all those articles, you can see that during this procedure itself, during the transplantation, many a times the donor itself die. Okay, that's really very sad actually. So there are many other reasons like because of which it's not possible, and plus it's very costly. Now why it is costly? Because of the maintenance, like stem cells, all these need high maintenance, like high, I want to say, contamination free area, and the media it is required, and to maintain them, everything, it's like high maintenance, and because of which it becoming costly. Okay. Now coming to the pluripotent stem cells.
Why anesthesia is not given during this process?
So because it is going to what to say, the stem cells loses its property because of the anesthesia. That is what I have heard and read about it, and I've seen also, like where I have gone to one of the hospital, and there this stem cell was going on. I didn't know that that time it was stem cell therapy was going on. See, I mean, you won't understand actually. So they take a very broad needle. Okay, it's just like a syringe only, but the needle is very broad. It's not very narrow, that it's easy to pierce. So it was directly pierced from the thigh, and it was directly gone into the bone, and from there it was extracted. So that was really very painful for the donor, of course. And my same question was that, like, why anesthesia was not given. So they said that it interferes because it goes into the blood, so it might interfere with its activity, like the stem cells may lose its property. So that's why. Okay. Yeah. Now, while adult stem cells are difficult to isolate in culture, it is relatively straightforward to isolate and propagate the stem cells of early embryos. See, even this is there. Now, adult stems, if you harvest and you try to culture, it's not necessary that out of the body it is going to, propagate in the culture, in the dish culture, right? In which you're going to provide the media. It's not necessary. Sometimes it really does not grow itself. It's not only about the adult stem cells. Even if you start working on the primary cell lines, like let's say that you have taken the liver, small piece of liver is taken, that liver tissue is actually homogenized, and you have taken the cells, right? Now the cells are suspended and you put into a very rich media of culture and all, everything you are doing, but it's not necessary that it is going to grow. It might die. So stem cells so or be difficult ho jata because they have got different requirements. The presence of the proteins and the transcription factors, everything is different and very difficult for the adult cells to be, cultured in a lab. So it's better to go with the embryonic stem cells. So how this is done? So these cells can be grown indefinitely as pure stem cells population, and give rise to all types of differentiated cell. Okay. Now, there has been enormous interest in embryonic stem cells, but here the thing is that ethical issue aa raha hai thoda sa. Okay, I'll tell you why. Okay, so first we have taken the embryo. So you know how the embryo is formed, right? So after the fertilization, your embryo starts forming. What you do is you extract those embryos from the womb of the organism. Now they can be propagated in the culture, and they can reintroduce into the early embryos and enable give rise to cells and tissues of the mouse. So what they're saying is that आप fertilize करो egg and sperm, fertilization is happening, it forms an embryo, right? Take this embryo, introduce your gene of interest, which is going to cure that particular disorder, then put it back. So because of this procedure, since this is against the nature, taking out the embryo, reintroducing certain other DNA into that embryo, and then again inserting back, right? Not inserting, sorry, implanting back. So it is unethical. So because of these, many people are not, in favor. The government is not in favor. I'll show the procedure here. Yeah. So as you see, so after what happens, the embryos are taken, right? This inner blastocyst stage where after the fertilization, the embryo starts growing, right? So at this stage, they take this inner cell mass. Now this cell mass can be cultured, and depending on the type of the transcription factor or, the growth factors which you provide in the media, these particular embryonic stem cells can be directed towards the one particular type of differentiated cells. Did you get it? So this is possible to study and, to do the research using mouse embryo. But with the human embryo, many ethical concerns are coming, and that's why it is not there, so popularly in the market.
Ma'am,
Huh.
can you repeat it, ma'am?
Okay, yeah. So here, see, I'll explain from here itself. So what we are doing over here is that embryonic stem cells, we can make use of it. Now, it is not that difficult to harvest. Now, why it is not difficult? Because see, let's talk about the mouse case itself, because in the humans, it is unethical. Okay. So in the case of the mouse, after the fertilization, okay, the egg and sperm fertilizes, your embryo starts forming. Okay. Now, at the blastocyst stage, where you have got this shape of the embryo, you take the inner cell mass. These cells are known as the inner cell mass. Inner cell mass. Now, this can be grown in the culture. What you're doing is that if you have your particular gene of interest, okay, let's say that this gene is responsible for one particular type of disease. Okay, let's say SCID. Okay, severe combined immunodeficiency, in that you don't have the AR gene. Okay, it's not AR, what is it? Adenosine deaminase. Adenosine deaminase enzyme is missing. So let's say that this gene of interest is that particular enzyme. Okay. Now you also need drug resistance marker, so that you can, isolate the modified or the transformed cells only. Right. So what you're going to do is you're going to insert this plasmid into over here. Okay. Now the sequences, the embryonic stem cells carrying the plasmid sequence gets integrated into the chromosomal DNA. So what we are saying, inner cell mass is there. Now your gene of interest is going and getting incorporated into the chromosomes of your embryonic stem cells. Okay. Now, only which has got this gene and the marker will survive in that particular drug containing media. Is this much clear? The transformed embryonic, only the transformed embryonic stem cells will be taken. Okay. Now it is inserted back into the foster mother. जिससे लिया है हम उसमें वापस नहीं डालेंगे. The other mother. Okay. So it is transferred to the foster mother, or we can say the surrogacy. Then what you see is that after so many generations, you are going to have the chimeric offsprings are produced. Features of this, and whatever the changes you have made, you are going to get, not feature of foster mother, sorry, not like the changes you have made and the mother, from where you have taken the inner cell mass. So what you're saying is that if you mate to the normal mice, okay, then it can go back to your the cell which you have got. Now why? Because of the homologous recombination. Now, here any steps which you didn't understand. So this is how embryonic stem cell or the therapy can be done. Okay, gene of interest you can put. But all this is easy to say, but very difficult to perform, because each step requires so much of the technicality that, most of the times it's failing, extracting, getting the inner cell mass, making sure that it is getting cultured in the media in which you have provided. Now the next, whether your DNA, this plasmid DNA is stable or not, whether it is getting transformed or not. If it is getting transferred, then are you able to successfully implant into the foster mother or not? Whether the foster mother is surviving, it is alive, so that it can mate and produce the chimeric offsprings. And then again, just to see whether the gene of interest which you have inserted is getting expressed or not. Okay, so at each step there are so many challenges. So it's easy to say, it's easy to explain like how the embryonic stem cell therapy can be done, but at each stage, as you can see, it is very challenging. Okay, and in most of the cases, like still the research is going on. Why, if you ask me like why it is failing, then the simple answer is that till now we don't know all the requirement of the proteins or the transcription factor which is stimulating your embryonic stem cells you to get differentiated into that. A body ke andar in the nature how it is happening, even though we have learned a lot, but still there are few proteins or the biomarkers or the genes which is being not studied yet. Okay, so even though we have got full science and technology with us, still there are few what to say, फिर भी कुछ चीजें हैं which is lagging behind, and because of which till now we have not reached that 100% completion. Okay, the same thing what we have discussed, and later you can differentiate into them different types of blood cells, epithelial cells. So all this depends like what kind of transcription factor, the growth factors you are giving. Okay, so the undifferentiated embryonic stem cells are there, then you give them the transcription factors. Okay, you give them the growth factors so that they get an idea on which path they have to undergo. So either they can form blood cells, epithelial, adipocytes, smooth muscle, nerve cells, or the heart muscle cells. Same thing. For example, methods have been developed to direct the differentiation of both mouse and human embryonic stem cells into cardiomyocytes and various types of neurons. So they are there until now. I can tell you right now also in the Cambridge University, one of my like BSc classmates, she was there, she was doing her project like intern. She went for MS over there, and there she has mentioned that she used to meet this Parkinson people. Not sorry, Parkinson, I'm saying Alzheimer people. Okay. And every day she goes and meets them, and every day they forget. And they were undergoing this stem cell therapy. So they have given those, injections of new neurons and everything. But still, since that age is so weak, it is not possible for the body to get used to it. So it was not that successful. So sometimes she saw that there is an improvement, that yeah, he was able to remember her in 2 3 meetings, but at the fourth day then again back to the same, like nahi pehchan raha hai, not able to remember. Okay, so it wasn't conclusive, like according to her, what she has mentioned to me was that it wasn't that conclusive, because the drugs, you have to give the drugs also so that the body doesn't reject, and it was painful procedure. When it is a painful, you have to give painkillers as well, and the medicine to heal. So all these combinations of drugs are making difficult for a body to adjust with the introduction of the new neurons or new cells or whatever it is, and plus the introduction of so many drugs to a weak body. So that's why it's failing. Okay, I'm not saying that it's a failure, but the completion or what to claim that we have got the cure for Alzheimer is not true. Okay, it's not 100%. Okay, so guys, I could prepare this much only. I'll be stopping here.